Today we are going to take the topic spermatogenesis. Spermatogenesis is the process of formation of the sperms. Sperms are formed in the seminiferous tubules of the testes. The lining of the seminiferous tubules is known as the germinal epithelium. It has got the germ cells, these are germ cells. Spermatogenesis occurs on the onset of puberty in males and it continues lifelong. It's a long and slow process and it takes nearly 74 days for the formation of the sperms. So let us take one of the germ cells. If we take this germ cells, this is one of the germ cells. This is the germinal epithelium. To understand the whole process of spermatogenesis, let us consider one germ cell. These germ cells are diploid in nature. So they are diploid in nature. Now these germ cells undergo mitotic division just to increase in number. So two cells are formed here and both the cells are diploid in nature. Now these two will again undergo mitotic division and form four cells. Now some of the cells, they keep on dividing. They are known as stem cells. Suppose we take this. These cells, they keep on dividing to increase in number. Now this acts like a stem cell and it is called as A-type cell. And some of the cells, they stop dividing. They are known as B-type cell. They are also called as progenitor cells. You can call them as progenitor cells. Now B-type cell, they will stop division. They will accumulate nutrition from the nutritive cells and grow bigger in size. That is, they are preparing themselves for mitotic division. So let us take one cell, one B type of cell. It will grow larger by accumulating nutrition. Till here, it is multiplicative process. Till here, it is when the cells are increasing in number. It is called as multiplicative phase. The cell here, the B type of cell, increases in size. It absorbs the nutrition and increases in size and forms the primary spermatocyte. So this is known as primary spermatocyte. Primary spermatocyte. This is diploid in nature. This is 2N. Now this cell, the primary spermatocyte, will undergo the first mitotic division. It has assumed lot of nutrition and is in a resting phase. That is, it is preparing itself for mitotic division. So it undergoes the first mitotic division. The first mitotic division. This is first mitotic division, and it forms two haploid secondary spermatocyte. Secondary spermatocyte. The formation of the primary spermatocyte, that is when the cells they absorb nutrition and grow bigger in size, this comes under the growth phase. This we can call as growth phase. So this is growth phase. And the last phase is the maturation phase. So primary spermatocyte undergoes the first mitotic division. This is the first mitotic division. And it results in the formation of two haploid secondary spermatocyte. Now these secondary spermatocyte will also undergo the second mitotic division. This will undergo second mitotic division and result in 4 haploid cells. This is second mitotic division. It undergoes and it results in 4 haploid cells, which are the spermatids. These are known as spermatids. Now you can see one cell has resulted in 4 cells, 4 haploid cells. That is, the primary spermatocyte, which is diploid in condition, underwent the first mitotic division and resulted in the formation of 2 secondary spermatocytes, which are haploid. The 2 spermatocytes, that is the secondary spermatocyte, underwent the second mitotic division and formed 4 cells, which are now called as spermatids. These spermatids are round in shape and non-motile. So the formation of spermatids from the germ cells is known as spermatogenesis. So till here it is spermatogenesis. The secondary spermatocyte, which undergoes the second mitotic division and results in 4 haploid cells, that is spermatids, this will come under the maturation phase. This is known as maturation phase. So I am writing it here. This whole is known as maturation phase. This whole process, that is formation of spermatids, the non-motile round structure, the spermatids from the germ cells, is known as spermatogenesis. This whole process is known as spermatogenesis. So we can see the round non-motile spermatids are formed. Now these spermatids will undergo morphological changes and convert into motile spermatozoa. So this is known as spermiogenesis. This whole process is known as spermiogenesis. In this process, certain changes takes place in the sperm cell. The Golgi body, it gives rise to acrosome. Acrosome is present in the head of the sperm cell. It contains an enzyme known as sperm lysis, which helps in dissolving the walls of the ovary during fertilization. There are several mitochondria arranged spirally in the middle region of the sperm. This mitochondria actually gives energy to the tail, and there is a tail, motile tail, which helps in locomotion. After spermiogenesis, the sperm cells, they cling together, they adhere to the Sertoli cells to obtain nutrition, and then they are finally released from the seminiferous tubule. So this whole process is known as spermiation. This whole process is known as spermiation. We have seen three processes today. First is spermatogenesis, that is the formation of the sperm cell which takes place in the seminiferous tubule, and then second is spermiogenesis. Spermiogenesis means the morphological changes that take place in the spermatids. Spermatids gets converted into spermatozoa, and then spermiation, that means the release of the developed mature sperms from the seminiferous tubules.